so we can start. So welcome to another Cancer Research UK RadNet City of London seminar series. I'm Aleksandra Vancevska and I'm a postdoc at the Francis Crick Institute in the lab of Professor Simon Bolton, and I will be your host today. So firstly, I just want to highlight the importance of the seminar series, which is intended to initiate connectivity and give insight into the latest and cutting-edge radiation research in both clinical and non-clinical areas, and hopefully offer you the expertise and a platform to build collaborations. Also, I will just go briefly through some housekeeping points. So please, if you can, mute your microphones and turn your cameras off while the talk is going. I encourage you to keep your questions until the end and raise your hand so that we can be more interactive. But if you want, also you can just put your questions in the chat box and we will answer them in the Q&A session at the end. And as you have noted, the meeting will be recorded. And without further ado, I would like to introduce our speaker today, Dr. Ester Hammond. She's a professor of molecular cancer biology at University of Oxford, and she will give a talk on the topic of targeting the biological response to radiobiological hypoxia. I personally think that this is a highly interesting and probably very cutting-edge research, because we know that there are parts of tumors that are radiation resistant, and these parts are mostly hypoxic. And another interesting part and another interesting crossroad that I think Esther is sitting with her research is that she's studying both tumor hypoxia as well as hypoxia-induced DNA damage, and also trying to understand how these lead to tumor resistance. So without further ado, I really want to thank Esther for accepting to give this talk, and I'm actually very curious and very interested to hear what she has to say. So welcome, Esther.
Okay, thank you. Right, it's my turn to demonstrate the wizardry. Okay. Okay, does that look good?
Yeah.
Brilliant. Okay, thank you so much, Aleksandra, for that introduction and for this invitation to present some of the work that's been going on most recently in my lab at the University of Oxford. So, as Aleksandra mentioned, my interest is tumor hypoxia, has been for many years now, and I'm sure there's no need, but just to catch everybody up, just in case, hypoxia is a common feature in most solid tumours. So most solid tumours will have some degree of hypoxia. It will vary tremendously, but they will have some. And of course, this occurs because there's a mismatch between oxygen supply and demand. So in the sections you're looking at here on the left, the brown staining are hypoxic regions, which you can see are a distance away from the blood vessels shown in the middle. And this is why hypoxia occurs. It's because these cells closer to the vessel have metabolized all of the available oxygen, leaving none out here. And of course, the reason that we care about this is that hypoxia predicts for a poor prognosis. And I think it's important to mention, although obviously we're joined by our interest in radiation, it actually predicts for a poor prognosis irrespective of therapy type. So however a patient is treated, the more hypoxic their tumour, the worse the prognosis is for them. The other thing that I like to introduce when getting started in one of these talks is the concept that hypoxia is not just one thing. It's not on or off, and it is in fact oxygen dependent. So different levels of hypoxia lead to different effects in the cell, different biological outcomes. And also to introduce the terms that I usually use. So normoxia is this horrible word that we use to imply normal, whereas what we actually mean is the oxygen concentration cells experience in tissue culture, which is of course in no way normal. More relevant is probably Hypoxia. Now this is the normal oxygen concentration that cells experience in the tissue of origin. And then we get into hypoxia and radiobiological hypoxia. So it's important to remember that this is not just one thing. And that brings me to highlighting some recent work that's going on in my lab, which I'm not going to talk about today, but I just wanted to show you a sneak peek of, where we're trying to develop probes. And at this point in the project, these are fluorescent probes, which will allow us to tell the difference between these different types of hypoxia. So to use these probes to highlight what the actual level of oxygen is that the cells are experiencing. So for example, probe 1 starts to give us a signal once oxygen gets down to about 2%, whereas this probe really doesn't get going until about 0.5% or lower. So these tools are going to be very useful for picking out these differences which occur. Now, of course, my lab is really interested in this level of hypoxia shown in the blue, the radiobiological hypoxia. And that's because, as you've already heard in the introduction, this is the level of oxygen where cells become significantly more radiation resistant. And that, I think this is the easiest way and most simplest way to demonstrate that, where we've taken a cancer cell line and irradiated it with these doses of radiation at these levels of oxygen. And what I hope you can all easily see is that the cells down at 0.1% oxygen, so this radiobiological level, are much more resistant to radiotherapy than those that were irradiated in 2%, so the blue line, which is not hugely different from 21%, that normoxic condition. So this radiobiological level of hypoxia, that's really where my interest lies and where my lab is focused. And our overall goal is to try and understand the biology and the signaling of cells exposed to this level of hypoxia, with the ultimate goal of finding ways to target them. We want to find something that we can inhibit or exploit that occurs at this level of hypoxia, so that we can make them as radiation sensitive as the cells in blue or red. So the way that we've been doing that is by investigating the DNA damage response, abbreviated to DDR here. And this work began many years ago. And the first thing that we found was that this level of hypoxia, radiobiological hypoxia, induces a DDR, which you can see here. You see these markers that you're probably all familiar with of the DNA damage response showing up at the 0.1% oxygen, but not the 2%. So again, this is another example of hypoxia is not all the same. This only happens at this level. But most interestingly, this appeared to happen in the absence of DNA damage. So this is a COMET assay. The hypoxia here is this radiobiological level, and you'll see there's no increase in DNA damage despite the presence of this signaling. And that was initially quite confusing. So obviously we did all the assays at our disposal over the years to try and work this out. And the conclusion is the same. We haven't been able to detect DNA damage in these conditions. What we have been able to detect, however, is replication stress. So again, probably most simply shown here, where the cells have been exposed to either hypoxia or neocarcinostatin. We see RPA forming foci very quickly, that's shown here, in response to hypoxia, indicating that we've got a replication problem. Further supported by DNA fiber studies, which show that the rate of DNA synthesis is significantly slower in hypoxic conditions compared to the normoxic. And again, here's our COMET assay showing no damage. Here's 53pp1. You see the nice nuclear foci you'd expect in response to neocarcinostatin. We didn't see any foci in response to hypoxia. Somewhat less clear when we look at gamma H2AX, we see the foci with the DNA damaging agent, We do see phosphorylation of H2AX in hypoxia, but we get this pan-nuclear staining. So this is much more indicative of replication stress as opposed to the occurrence of DNA breaks. So we are confident that this level of hypoxia leads to this rapid replication stress response. And as I said, the reason we're doing this is to try and tease out parts of this response which could be targeted in order to increase the radiation sensitivity of these cells. And one example of where we've been successful doing that is by targeting the ATR kinase. So here on the left, this is a colony survival done in hypoxic conditions, plus and minus the ATR inhibitor in combination with radiation. And you can see we've brought those radiation resistant cells right down and increased their radiation sensitivity by inhibiting ATR. And this we were able to show in an in vivo model as well, where you're looking at these red spots is the combination of radiation and ATR inhibition. So this supports our hypothesis that, by thoroughly and, investigating the hypoxia-induced replication stress, or DDR, we will identify potential therapeutic targets. So how do we go about that? The first question we asked is, well, why have we got this replication stress? Why is it that within, no more than a couple of hours of putting the cells in the chamber, we've got all this single-stranded DNA and replication forks slowing right down? So we really thought about, what could be the causes of this, and, used this diagram as a place to start, taken from this lovely review. And the first thing we investigated is shown over here, limiting nucleotides. And what we found was that when we measured DNTPs in hypoxic conditions, we saw a very significant and rapid drop in DNTP level. So that this simple graph here is showing you that all 4 nucleotides decrease in abundance. And this time when it's just 3 hours, which very much correlates with that change in DNA replication rate that we also see in these conditions. So this immediately suggests that one of the problems in hypoxia is a lack of nucleotides. And I'm not going to go through the work we did to understand this further fully, other than to just show you this slide, which is taken from the paper we subsequently published. Because what we found was that in response to hypoxia, the enzyme ribonucleotide reductase, which is essential for the de novo synthesis of DNTPs, is significantly compromised in hypoxic conditions. And specifically, the small subunit RRM2 really struggles in the absence of oxygen. This leads to replication stress. One of the consequences of that replication stress is to turn on the DDR, as I've already shown you, which does include P53. P53 then turns on the alternative small subunit of RNR, which is RRM2B, which is able and appears to work more efficiently in hypoxic conditions. So we think that it's this switching that is responsible for the low levels of DNTPs that we see in hypoxia, which in turn probably contribute to this lack of damage that we see. But this is, as I say, this work is published, so I'm not going to go through that more today, other than to say that we're confident that one of the causes of replication stress in hypoxia is this lack of nucleotides. But we also felt that it was unlikely to be the only thing that was going on, that we thought that there were probably other contributing causes of the replication stress in hypoxia. So we went back to the diagram, and this time we focused on this other end, and specifically the R loops, the presence of R loops. So R loops are stable hybrids between RNA and DNA, shown schematically here. They can be, the thing that we found particularly tricky about R loops is how to measure them. So I'm just showing you here on this schematic how we've gone about that. So one of the tools most commonly used, which I'm sure many of you are familiar with, is this antibody S9.6, which specifically recognizes these hybrids between RNA and DNA. The other tool that is tremendously useful is the overexpression of the enzyme RNase H1, which will degrade the RNA in the R loop, which means in your experiments you can always take a plus and minus RNase H1 approach, and if something is R loop dependent, should go away if you treat them with RNase H1. There's also a mutant of RNase H1, this D210 mutant, where it binds the R loop, but then is unable to disengage from the R loop. So this is another useful way of being able to measure R loops and quantify them, because we can tag this and measure the signal that we see. So having got these assays up and running, we asked the very simple, what happens to R loops in hypoxia? And what we found was that this is using the S9.6 antibody, that actually R loops accumulate in hypoxic conditions. That's this condition here. And importantly, you can see we've used the RNase H control, and the signals go away when you use the control. And shown also here is a picture of what that immunofluorescence looks like, and also points out that we can use this antibody in a slot blot as well, although we prefer the IF assay. To back that up, we obviously used our other assay with this RNase H1 mutant. And again, we saw the same thing, that R loops are accumulating in hypoxic conditions. So this was, we were looking for a source of replication stress. We'd already found a lack of nucleotides. Now we appear to have R loops accumulating in hypoxia. Doesn't mean they're significant. So we did a range of experiments before going too much further, just to try and get a sense of how much of these R loops actually contributing to this replication stress, or DDR, that we're seeing in hypoxia. And I'm just going to go through this data quite quickly. The first thing we saw in A is that we could reduce the DDR by decreasing R loops. And here we've used that trick of overexpressing the RNase H1. So where you see a plus, it means there are less R loops. And you can see the DDR markers are decreased when you get rid of the R loops. We saw that some of the hypoxia-induced chromatin changes that we're used to looking at were also affected by the loss of R loops. So you see here, the H3K9 di and trimethylation are decreased if you get rid of the R loops. Perhaps not surprisingly, the markers of replication stress are also decreased. You can see this difference in RPA phosphorylation, CHEK1 phosphorylation. We can also look at that another way by looking at the cells with RPA foci. We can bring that down by getting rid of R loops. And indeed, we can speed up replication by getting rid of the R loops. So this was enough to convince us that these R loops, they were there in hypoxia, and also they were having an effect and seemed to be contributing to the DNA damage response we see in these conditions. But as exciting as that was, it did cause us to scratch our heads for a little bit, because if we think about what an R loop is and then ask why would they be accumulating in hypoxic conditions, it's not immediately obvious, because just as quick as the DDR being induced in hypoxia, we see a decrease in global transcription levels. So this is an EU incorporation assay shown here and then quantified here. And it's when you put cells in hypoxia, they quickly turn off transcription. And of course, you need transcription for R loops. So it's not, as I say, it was not immediately obvious to us why they would actually be accumulating. So with a view to trying to understand that, we, in a very unbiased way, thought, right, well, who are the players in the cell that control R loops? How do they accumulate? Who resolves an R loop, etc. And realized that there were a lot of factors involved. So we looked at the expression of some of these factors that we know are key to R loop resolution. And shown here in the top left, this is just a qPCR looking at expression of R loop associated factors in hypoxic conditions. And the dotted line is where they are in normoxic conditions. And there's two things to notice. One, and this is obviously only some of the factors that we looked at, but the vast majority of everything we looked at was repressed or decreased in hypoxic conditions, which, you know, might not be surprising. I just told you that transcription goes down in hypoxia. However, this one, Senataxin or SETX, stuck out in that not only was it not decreased in hypoxia, but it actually accumulates in these conditions. And so this got us really excited, and the rest of the data on this slide are, you know, the things we did to convince ourselves that this was real. So first of all, we looked at the oxygen dependency. So bottom left, you see Senataxin is going up with time in hypoxia, but this is in radiobiological hypoxia. So that level where we see the DDR, it's not changing at 2%, which is definitely hypoxic. There's plenty low enough to give you a HIF response, but no change in Senataxin. We could also see this at the protein level. And I should mention, we saw this in several cell lines. The other thing that we were able to do, which convinced us that this was real, was that we saw a correlation between Senataxin expression and hypoxia-induced p53 target genes. So p53, again, is only active in radiobiological hypoxia. And we have previously characterized a number of genes that p53 turns on in those conditions. And that's what we've correlated. So Senataxin with the targets of hypoxia p53. So this gives us confidence that this happens and that it happens in patient samples. So we found Senataxin looks interesting, it's going up. What does it do? And, you know, I haven't got time to go through all of the things Senataxin has been shown to do. It's got many functions. But of course, the one that stands out is that it's known, it's an RNA DNA helicase, or at least a putative RNA DNA helicase, and so it can unwind R loops. So this leads to the very obvious hypothesis that, you know, loss of Senataxin should increase R loops even more in hypoxia. So I've already seen that R loops are going up in hypoxia. I now know that Senataxin is going up in hypoxia. If I take the Senataxin away, I expect to see more R loops. And that is exactly what we saw. And again, I'm just going to go through this data very quickly because it is now published. So when we took Senataxin away, we saw the R loops go up in hypoxia. And this looks complicated because of all the RNase H controls, but it is that simple. When we took Senataxin away, we slowed down replication rates. So replication went even slower, which, you know, because we believe we have increased the R loops. We also found that when we took Senataxin away, we saw an accumulation of DNA damage in hypoxia. So remember, we weren't seeing DNA damage in hypoxia alone. Now, if we take away Senataxin, we do. And this loss of Senataxin in hypoxia increases the cell death that we see in hypoxia. So we see more cells dying if they don't have Senataxin at this level of hypoxia. So this is obviously very interesting to us, but you know, clearly we've been surprised to see Senataxin going up in hypoxia, and we wanted to know how. What was the mechanism? Who was turning on Senataxin in hypoxia? Because we've seen that it's transcriptional. So the first thing we did was rule out the usual suspects. So this is not HIF1 dependent or HIF dependent, and is not dependent on p53. I'm not showing you that data today, but I can tell you we thoroughly ruled that out. The data that caught our eye was this data. So this is where we've just taken cells and exposed them to different stresses to see which ones turn on Senataxin. And as I've shown you previously, radiobiological hypoxia turns on Senataxin, but so do these two agents, tunicamycin and thapsigargin. And for those of you who are not familiar with tunicamycin and thapsigargin, or what they are commonly used for, they are commonly used in the lab to turn on the unfolded protein response. And again, for those of you unfamiliar, this is a stress-responsive pathway in the cell, which is initiated by the accumulation of unfolded proteins in the ER lumen. And you know, the interesting thing here, which again you may be unfamiliar with, is it has the exact same oxygen dependency as the DDR in hypoxia. So when you take cells to radiobiological levels of hypoxia, in the same cell you will initiate a DDR, which we believe is downstream of a replication problem. You will also, in the same cell, have a UPR. And this is because the fold in making disulfide bonds requires oxygen. So the same cells will be having both of these pathways. And the fact that tunicamycin and fapsogagin turned on senotaxin was a very big hint that perhaps these two pathways meet at this point with senotaxin, and that the UPR could be turning senotaxin on. So here's that data shown just perhaps even more simply. So in hypoxia, we have single-stranded DNA turning on the DDR. We have unfolded proteins turning on the UPR. We can see markers of both of those on a Western blot. So in the green box are DDR markers, in the blue box are UPR markers. And just to remind you, because I think perhaps I didn't mention this earlier, the accumulation of R loops also fits with this oxygen dependency. So we see the R loops accumulate here in the dark blue, which is radiobiological hypoxia. We don't see them accumulate in the paler blue, where it's 2% oxygen. And sure enough, by looking very carefully at the senotaxin sequence and a bit of trial and error, we found that senotaxin expression in hypoxia is dependent on ATF4. So this is plus and minus ATF4 and a qPCR. This is an ATF4 chip, so that we can see ATF4 accumulating at the senotaxin promoter in hypoxia. And ATF4, or rather the PERK kinase signaling to ATF4, is one of the three main branches of the UPR. So we're convinced that senotaxin is induced through the UPR. Now this was particularly exciting because, as I mentioned, ATF4 is a transcription factor that's regulated by PERK, which is a kinase. Kinases are easily inhibitable. So this raised the possibility that we should be able to control senotaxin level and therefore R loop accumulation in hypoxia by targeting PERK. And sure enough, that is exactly what we found. And the data you're looking at here is a mixture of both siRNA and PERK inhibitors. And I'm not going to go through this because basically we found the same thing. All of the things we saw when we took out senotaxin, we were able to recapitulate by targeting PERK. So this has led us to this model, which has now recently been published, whereby in hypoxia, we see an accumulation of R loops leading to, and this contributes to the replication stress response. And we see this parallel pathway, the UPR, working through PERK signaling to senotaxin to try and restrict this. So if we take out any part of this, this all gets much worse. But if you remember at the beginning of the talk, I said the whole point in what we do for us is to try and understand this biology so that we can then think about targeting it. So obviously we asked, what happens if we take away senotaxin and then irradiate these cells? Have we reversed, can we reverse the radiation resistance? And there was some data already available from Martin Lavin's lab in the literature, which showed that actually patients with mutations in senotaxin, their cells, they weren't radiation sensitive. There was no difference in radiation sensitivity in these cells. These cells are lymphoblastoids, so we felt it was important to repeat this in our own hands. And this is the data that we have. So using an siRNA to knock out senotaxin, we've irradiated the cells, and we can see in both normoxia and hypoxia, there is an increase in radiation sensitivity when you take away senotaxin. Now, I'm not going to pretend that this is the ideal result. What I would really like to have seen is an increase in radiation sensitivity in hypoxia. and no effect in normoxia. These experiments are ongoing, and it's going to be key to investigate what happens in normal cells. Do we see this same effect in normal cells, or is this something we only see in cancer cells? So that is very much ongoing, but we're encouraged that we're on the right track, and that potentially there are more genes that are regulated by the UPR that have an effect on the DNA damage response in hypoxia, and that potentially this is a good source of future, of molecules to target in the future. So of course we've all done things that don't usually do as a result of the labs being closed. So one of the things that we did was do a bit of Google science. We downloaded some RNA-seq data, some CRISPR screen data from various other labs, and used it to try and identify genes which are both induced by the UPR, they're induced in hypoxic conditions, and have been shown to cause a change in sensitivity to DNA damaging agents, including radiation. So we did this. We ended up with 103 genes in the middle that have been classified this way. Interestingly, 83 of them have been classified as non-classical DDR genes. And we are working our way through this list to see if there are any potentially interesting targets there. And I should say we were very pleased to see during this analysis that Senataxin did indeed come out in this list. So we're encouraged that we're on the right tracks. So far I've told you a small amount about nucleotide levels and how this leads to stalled replication forks in hypoxia. More recently, we've also shown that there are R loops accumulating in hypoxia. And what these obviously both have in common gets back to one of those very first slides I showed you, which is that these cells in hypoxia have a lot of single-stranded DNA in them. This is so quick and so obvious in these cells that it led us to ask, well, what happens to this single-stranded DNA, and is it vulnerable to cellular processes, which in turn might have an impact on genomic instability? So I've told you a little bit about how it occurs in hypoxia. I've shown you that it does. I've told you how I think it accumulates in hypoxia. I now just want to talk a little bit about one of the things that we think can then happen to that single-stranded DNA. And it was this thinking that led us to investigate the APOBECs. So the APOBECs are a family of deaminases. There are 11 of them, highly conserved enzymes, which deaminate DNA. So they act on, although they can act on RNA as well, they primarily act on single-stranded DNA. So you can see why we were interested, especially that there are increasing number of reports showing that they can act on stalled replication forks and R loops. For those of you who haven't heard of the APOBECs and think that's a bit odd, why would you want something in your cell that can effectively mutate, change a cytosine to a uracil in single-stranded DNA? It is, of course, because probably their true function is to restrict invading viruses. They're trying to mutate viruses to stop them infecting your cells. But it has become increasingly apparent that this is relevant to cancer, and that has led to the development or the identification of the APOBEC signature, which is one of the well-characterized mutational signatures, and importantly is second only to the aging signature. So this is really common in human tumors. As shown here, the APOBEC signatures are the two in the red boxes. So this led us to ask, does the replication stress that we see in hypoxia lead to increased APOBEC activity, and therefore is that contributing to genomic instability? So we started very simply and looked at the expression at the mRNA level of all the APOBECs that we could detect in our cells. And I'm only showing you 5 of them. We were able to detect, I think, all but 2. And initially we were very encouraged. So you see in the orange bars are the cells that are exposed to radiobiological hypoxia. There is also a milder hypoxia, so 2% on these graphs. And we used hydroxyurea as our positive control because a very nice report in the literature had already told us that this would lead to APOBEC expression, or specifically APOBEC 3b. So as I say, we were initially quite encouraged. The orange bars are generally bigger, but actually closer examination, we weren't so pleased because this one on the end is APOBEC 3b. And you can see it's actually going down in radiobiological hypoxia. And the reason that this sort of tempered our enthusiasm is because along with APOBEC 3a, this is the one that's been shown to be the real driving force of that mutational signature in human cancers. It's also expressed in the nucleus as opposed to pancellular. And therefore, this is the one that has access to that single-stranded DNA, which I hypothesized might be vulnerable to APOBEC attack. So not daunted by this, we thought, well, this is just qPCR. Who knows what's actually going on? Let's do a functional assay. So we did exactly that. And that assay is very simple and shown schematically here. So we take a piece of DNA with a site that can be deaminated. The DNA is tagged. And basically, if you get deamination, then the substrate band breaks and you end up with a smaller product band, which then you can run on the gel. So that's what one of these assays look like. Here's one of the gels. Below is a quantification. And as you can immediately see, actually, it pretty much does mirror the expression data that we had. So deamination is going up with hydroxyurea, just as we expected, but it's decreasing in radiobiological hypoxia, and it's not changing in hypoxia. So we thought, oh, well, that's great. There's nothing wrong with, if you take a hypothesis and prove it's wrong, then that's equally valuable. Doesn't look like there's a link between hypoxia and APOBEC activity. But before we turned our back on this completely, we made use of our proximity to Francesca Buffer here in Oxford and just thought, what do the patient samples tell us? And actually what we found was that there was a correlation between the hypoxia signature and the number of APOBEC-mediated mutations. So using the hypoxia signature to pull out the most hypoxic tumours in the TCGA data sets, we could find that they were also the tumours with the most APOBEC-mediated mutations. We were also able to show that there was a correlation between the hypoxia signature and the expression of A3A and A3B. Remember, these are the two that drive that mutational signature. So the more hypoxic, this is telling us that the more hypoxic the tumour is, the more the expression of A3A and A3B. So at this point, we thought, right, well, we haven't, there's definitely a link here. We clearly just don't completely understand it. So we really took a step back and thought about the data we had, which was showing that hypoxia was not changing APOBEC activity, but hydroxyurea is. And we often think of them as very similar. So this really confused us for a little while. So as I say, we went back to basics and thought about what are the actual differences between exposure to radiobiological hypoxia versus hydroxyurea. And these are the two things that we sort of zoomed in on, which was that, as you'd expect, if you put cells in radiobiological hypoxia or expose them to hydroxyurea, you see a decrease in DNA synthesis. But actually, there is more synthesis going on in the hydroxyurea-treated cells compared to the hypoxic ones. So there is still some, we have a number of ways of demonstrating this, but shown here very simply, these cells are incorporating more BRDU with hydroxyurea than they are in hypoxia. We also took another look at the DNA damage, and whereas again, using all of our markers of DNA damage, if you put cells in radiobiological hypoxia, you don't see an accumulation of DNA damage. We did see this with hydroxyurea. We did see DNA damage. So we wondered whether these differences were key, and is this giving us a hint as to why why two stresses that we think of as being similar are leading to two different effects, i.e. one's giving us APOBEC and one isn't. And also, is this going to help us work out why in the patient samples we are seeing this link between hypoxia and APOBEC activity? And we should have listened to ourselves, really, because what we started to think about is, of course, hypoxia is not just one thing. Cells don't become hypoxic, reach a certain level of oxygen, and just then sit there only experiencing that level of oxygen. Far more common is that cells cycle between different levels of oxygen. And this can be for a number of reasons, including blocked or occluded blood vessels closing and then opening. So at 1 minute there is no oxygen being delivered and the next minute there is. So we have to remember that this environment is very heterogeneous and that cycling hypoxia occurs. And so this suggested the hypothesis to us that maybe this was what our patient sample data was telling us, and that maybe cycling hypoxia was leading to APOBEC activity. So to test this, the first thing that we had to do was come up with a model for cycling hypoxia. There is quite a literature on this, and importantly, there's quite a literature that shows that the response to cycling hypoxia is different to chronic hypoxia. So you don't just see the same things happen in the constant levels of hypoxia if you then cycle. So using the literature as a guide, we came up with these conditions. So the cells are exposed to radiobiological hypoxia, followed by the milder level of 2%, which is still hypoxic, still a nice HIF response here, and then they cycle back. And obviously we can do this as many times as we like. And one of the things, in researching all this, one of the things that obviously piqued our interest was that cells exposed to cycling hypoxia have been described as having higher radiation resistance. So as soon as we had this model up and running, we decided to check that out for ourselves and put the cells either in constant 2% or in the cyclic conditions and then irradiated them. And obviously the key thing here is that in order to be able to compare the pink and the green lines is that they were irradiated at the same level of oxygen. So they were both irradiated at 2%. And you can see there is a significant difference. So just like the literature tells us, cells exposed to these cycling conditions are more radiation resistant. So a little bit more about our model. First thing we had to check was, in cyclic hypoxia, do the cells still experience replication stress? So we used a number of assays at our disposal. So as you'd expect, we still see phosphorylation of CHEK1 in cyclic conditions. We still saw the RPA accumulate in cyclic conditions. We could see a nice correlation between RPA and gamma H2AX in the cyclic conditions. And most importantly, we saw that in the cyclic conditions, we have some evidence of ongoing DNA replication. So in contrast to the radiobiological hypoxia, where we see an almost complete loss of DNA replication, and obviously, this goes with the fiber data as well. In the cyclic conditions, we see that the cell cycle looks very different. We see some evidence of ongoing replication. And we are very intrigued by this, and just as a small aside, we are very much trying to work out what's going on in these conditions at the level of the cell cycle. And the approach we're taking is to use mathematical modeling for this. This is some excellent work done by Julia in the lab, who is going to try and model the cell cycle response to these cyclic conditions, because we really think this is the only way we're going to be able to do this, because otherwise it involves changing those cycling parameters and doing FACS and then changing them a bit more, doing some more FACS, and we just feel that the number of experiments is endless. So we're hoping the modeling is going to be able to help us. But back to the APOBECs. We, of course, put the cells in our cyclic conditions and asked now what happens to the APOBECs. And to our pleasure, we saw that actually things were changed now. So whereas again, in the constant level of hypoxia, the APOBEC is not induced, shown here in IF and here on a Western blot. In the cycling conditions, we did see APOBEC increase just as we saw it when we treated the cells with HU. And of course, this is expression, this is functional data here. So here's the deamination assay. And again, deamination is going down in the constant hypoxia, but it's going up in cyclic, just like it did with hydroxyurea. So it seems to be that, you know, there is a link between hypoxia and APOBEC activity, but we had to change the hypoxic conditions so that they were cycling. And one of the things that we've already noticed about the cycling cells is that there's more replication ongoing in these cells. And that led us to propose this hypothesis that actually A3B is induced in hypoxia, but this is in cyclic hypoxia as a result of replication catastrophe. So we believe that it's not replication stress in these cells that leads to the DNA damage, but replication catastrophe. And what I mean by that is cells which are experiencing replication stress to such an extreme that they also accumulate DNA damage. And I'm just going to show you just a couple of pieces of data to confirm that to support that hypothesis. So first of all, if we reduce the DNA synthesis in hypoxic conditions and that the APOBEC expression is dependent on that, we should be able to reduce APOBEC expression and activity. And sure enough, by using a number of agents to block origin firing in these conditions, we were able to reduce DNA synthesis again, so it looks more like the chronic hypoxic conditions. And when we did this, we saw a decrease in APOBEC expression, and we have activity as well. So that provides a link to ongoing replication. We then looked for the presence of damage. And sure enough, in contrast to the cells exposed to chronic hypoxia, we saw an accumulation of 53 BP1 foci in the cyclic conditions. This is actually obviously an example of nuclear damage, but we were also able to see an example of DNA damage as detected in the cytoplasm by looking at the expression of cGAS, which was increased in the cyclic conditions, but not in the chronic conditions. So we have increased replication in cyclic hypoxia, which when we stop or prevent that, APOBEC expression goes away. We also have DNA damage in these conditions, which together suggest replication catastrophe. Now, one thing that I usually get asked, and there's usually someone, you know, very eagle eyes in the audience who spots the fact that these cells in the cyclic conditions are bigger than the cells in the normoxic conditions. And it was that observation that led us to carry out this experiment, where we've treated our cells with cyclic hypoxia as usual, but then we've allowed them to recover for 24 hours, and we've done that in the 2% conditions. And what we found is that during that recovery period, two notable things happened. Firstly, the DNA damage is persistent. It doesn't seem to be efficiently repaired. We can still see these foci at the end of this experiment. And also the cells have got even bigger. So I should point out that this cell line that we've done this analysis in is P53 positive, wild type. And therefore we are very much investigating as to whether these cells are senescent. But of course, you know, our cell line has wild type P53, most tumor lines don't. So it will be important when thinking about the fate of these cells to take P53 away as well. So in conclusion, what we found is that the APOBEC3B is not induced in response to hypoxia alone, irrespective of the level, but it is active in hypoxic conditions which include ongoing replication and DNA damage, and those conditions are cyclic hypoxia. And I'm hoping I haven't confused anyone, but perhaps you're thinking, didn't she start this by saying that using the hypoxia signature, you could show that patients which were more hypoxic had higher APOBEC expression, had higher levels of APOBEC-mediated mutations. How does that fit with APOBEC is not induced in response to hypoxia alone? And so it's just worth reminding you that the hypoxia signatures that we use primarily report on HIF activity. So these gene signatures, most of the targets, most of the specific genes are HIF target genes. And of course, these HIF is induced in all of the conditions I've shown you today, obviously not in normoxia, obviously, but you can see from this Western blot, there's HIF at 2%, 0.1%, and in the cyclic conditions. And this HIF is turning on its target genes, shown individually here, or 3 examples, and then shown here with the buffer score. So we think that that link that we've identified between APOBEC and hypoxia in patient samples is because the tool that we used to stratify the samples for hypoxia can't actually distinguish between these different types of hypoxia, which really brings me back to where I started, and to say that, you know, the development of tools to measure different levels of hypoxia so that we can tease out these different biological effects is absolutely critical, as is being able to measure what happens and how oxygen fluctuates in tumours. And I'm very excited by this collaboration that I'm part of with Sarah Bondig in Cambridge and Helen Byrne in maths, where we're going to try and do exactly that. So we're going to image blood flow in tumours. We're going to use that information to mathematically model how that will affect oxygen level in tumour cells. And I will then be able to test that, to model that and mimic that in the lab, and obviously ask how it impacts therapy response, because we know that it does make a difference. I've already shown you that it increases radiation resistance. So this is something that I'm very excited for the future. And with that, I will hopefully, I've had pictures of people throughout, and these are, you know, people who obviously actually did all this work, and I acknowledge all of them, and my collaborators and funders. And with that, I'd be happy to take any questions. Yes, I should stop sharing.
Thank you, Esther. I see a lot of questions, and maybe first the raised hand. So Sadhanand, if you want to ask the question.
Sure. Thank you so much, Dr. Hammond. Fantastic talk, and I got to learn so much from this talk. Thank you so much. Now, you provided some great data about Cytotoxin getting induced by ER stress. Is it UPR dependent? Do you see an endoplasmic reticulum stress element upstream of Cytotoxin, or is it just that fact that the DNA damage is leading to activation of PERK, and downstream of that, as the cells are dying or aging, Cytotoxin is getting activated?
I'm sorry, I'm not sure I completely heard the first part of your question. I think so apologies if I'm going off in the wrong direction, but the hypoxia-induced UPR response is very well characterized. So the 3 arms, one of which is PERK, is very much induced by this accumulation of unfolded proteins in the ER. We don't think that DNA damage induces PERK. Is that?
The cell death pathways also activate PERK, right? You know, when there's accumulation of there's multiple other pathways that also activate PERK. So maybe, you know, I'm just thinking out loud. If it is an ER stress dependent pathway, then you should have an endoplasmic reticulum stress element, because this is transcriptionally inducible, right? So typically all UPR inducible genes have this stress element upstream in the 5' UTR and upstream of the open reading frame. So, you know, an ERSC element or UPRE element would confirm that Cytotoxin is actually induced by UPR stress.
Okay, I'm sorry, I'm really struggling to hear you a little bit, but I think we were able to show using CHIP and by sequence, obviously sequence analysis, that the senotaxin promoter has an ATF4 binding site in the promoter. So, and we can see senotaxin induced whenever we turn on ATF4, however we do it.
Okay, yeah, maybe I'll follow up, via email.
Please do, because this is, this sort of intersect between the UPR and the DDR is something that's really fascinating us at the moment.
Very fascinating, exactly. Yeah, thank you.
Thank you.
So we have another question in the chat box by Jonathan. Really great talk, Esther, really informative. Do you think cyclic hypoxia triggers some impact on nucleotide synthesis? Are loops and the UPR senotaxin response unseen by persistent radiobiological hypoxia?
Oh, hi, Johnny. That's a really interesting question. So, to the same impact on... So, yeah, really interesting. The only one we've done so far, for sure, is the UPR. So we still see an accumulation of a GRP78 in the cyclic conditions. I don't know about nucleotides, and I don't know about R loops either. We are struggling a little bit with, when to measure. These cells are cycling, so you've not only got where in the cycle do you measure, but after, the usual length of exposure, how many cycles, but also where in the cycle. And for sure, I mean, that's, you'll have understood the radiation data, right? That's why it was so key that we're comparing it to stuff that was irradiated at the same oxygen level. But yeah, we will gradually work our way through that.
So that brings me to another sort of follow-on question. Are you guys still doing lots of chronic hypoxia exposures, or are you shifting away from that? Because obviously the relevance to what happens in patients is a little limited there, but it's easier.
Yeah, it's much easier. We're not moving away from it. I think what we have always been very careful to do, and we will continue to be careful to do, is that we keep our exposures very short. So, at the start of my talk, when I was talking primarily about senotaxin, those experiments, the longest time point's about 6 hours.
Okay.
Right? So we know full well that even if you've lost P53, if your cell's sitting at near as damn it anoxia for, 12 hours, who cares, right? You're going to die. So everything we study, I mean, some of those RPA foci are there in, I reckon you could catch them in an hour.
Right, okay.
So very aware of that, and yes, moving more towards the cycling, or at least not reoxygenating to 21%.
Yeah.
We're doing quite a lot of, let's expose it to radiobiological hypoxia for a short period of time, and then pretend that vessel just opened and turn it up to 4%.
Okay.
That's why I need the mathematicians, because I can't understand it otherwise.
It's very, very complex, yeah, and you're quite right. That dynamic thing it's difficult to predict exactly when you should switch it on or turn it off. It's complex. Okay, thanks, Esther. Great talk. Thank you.
Thanks, Johnny.
And the next one is Isabel Perez. You have it both.
Yep. So, hi, Esther.
Hi, Isabel.
Great talk. Really enjoyed it. It's really good to see on good days. So, my question is kind of following a little bit on from Johnny's and about cyclic hypoxia, but it's actually adding a little extra thing to think about. So in the tumor, obviously, when you put them in the chamber, you're just changing the oxygen tensions. But in the tumor, you might have flow issues and actually shear stress issues. So what do you think could be the contribution of that to your model?
I think there will be a contribution. I don't know what it is. But yeah, you're absolutely right that the shear stress, these physical kind of stresses, probably just as, if not more relevant, but couldn't predict which way it's going to go, I'm afraid.
Yeah. All right, thank you.
No worries.
And we have a question from Stephen. Thanks for a great talk. How do you think this is all related to radiotherapy resistance? Are there any other pathways you've seen that are active in response to cycling hypoxia?
Okay, that's another great question. We are we're very interested in the cell cycle response at the moment. That's where we're going at the moment to try and investigate that. We've shown that obviously cells exposed to radiobiological hypoxia are radiation resistant. We know why. One of the main reasons for that is because obviously you need the oxygen to form the double strand breaks that you rely on to kill the cells. The increased radiation resistance we see in cyclic hypoxia, we are unsure whether that is as a result of biological signaling, or whether it is a function of the change in the cell cycle populations, because obviously radiation sensitivity varies through the cell cycle. So we will be investigating, or are investigating, that first.
It's brilliant. Thanks very much for a great talk.
All right.
Maybe I can end with a question, because we don't have any more in the chat box. Sure. So one thing I have done, and I'm curious what you think about it. So I actually had a panel of drugs where I treated hypoxic cells at 1% and 0.1% of hypoxia over 5 days, and just did cell titer glow assays. And what is interesting for me is that I've seen this RPA foci increases, and there is replication stress, but for example, they're not sensitive to replication stress drugs. So I'm just wondering, how do you explain that? Because I can't, or can you?
So you see replication stress at 1%?
0.1.
0.1, okay.
Yeah.
But they're not sensitive to things like ATR inhibitors?
Not strongly to ATR inhibitors, and also not to acetylcholine, for example, which also blocks replication stress. So I mean, the only thing I'm thinking of is just the effect on the cell cycle, which then the replication is already slower. So I was wondering, actually, I had also, when I saw that data, I had this idea of like, well, if I put them back into normal oxygen, will then they be even more sensitive because they're primed in a different way? So it's just a little bit weird that they are replication kind of attenuated, but they are not sensitive to replication stressors.
Yeah, so I think if you look closely at the literature, particularly the literature published by Isabel Perez, you'll see that those assays involve a reoxygenation step. So they're not measured. You will see different things if you take your measurement at in the hypoxic fraction as opposed to after reoxygenation, which is why, going back to Johnny's point, we're moving away from reoxing all the way to 21 and trying to keep it a bit more physiologically relevant. But I suspect you will find, that's where you'll find your answers.
So I think we are done here. Thank you again for the great talk and for accepting the invitation.
That's all right, my pleasure. All right, have a nice rest of the day.
Yeah.